So this is another one of the concept bites, this time on emission and excitation fluorescence spectroscopy. So both techniques use exactly the same spectrometer, exactly the same. And for a steady state fluorimeter, that would consist of a source, and there would be a monochromator at the source. There's then a beam splitter which splits the beam of light into two. One of them goes to the excitation detector and the other will pass through the sample. The reason for this is that fluorescence depends on how much light is absorbed, and we therefore need to characterise this. The amount of light going in can vary, either with fluctuations in background voltage, or more particularly with wavelength of the source. If we're using different wavelengths, we want to make sure that we can normalise for that. So we've seen the xenon arc lamp characteristics. We know that it's not an even emission, and so the excitation detector is just basically used to normalise for that. The light then passes into the sample cell and we measure emission at 90 degrees. This is to eliminate as much of the incident radiation as possible. There'll always be a certain amount of Rayleigh scattering, and there'll always be Raman scattering, which is how we'd calibrate the instrument. But largely at this angle, the amount of direct source light passing through to my detector is minimised. Again, it passes through a monochromator before it hits the emission detector, and this gives me the ability to select both excitation and emission wavelength. There are principally two different types of fluorescence spectroscopy. Emission spectroscopy is the one that we're probably thinking of. This is, I have a fixed wavelength for excitation and I scan the emission. I scan the fluorescence spectrum. But I can also use excitation spectra. An excitation spectra is where I would fix the emission detector to a point where the molecule is fluorescing, but then I would scan the excitation source, almost as if I'm running an absorption spectrometer. And what I should see in the excitation spectra is something which, more often than not, is very similar to the absorption spectra. Excitation spectra gets very useful when there are differences between the absorption and the excitation spectra. And we can use this to look at energy transfer, as I'll talk about a little bit later on. The other thing to note in this is the presence of excitation slits. The excitation slits are used to control the bandwidth of light going into the sample and coming out. One of the problems with fluorescence spectroscopy is the fact that the emission intensity is dependent upon a number of things. It depends on how much the molecule is absorbing, which depends upon the extinction coefficient and is ultimately wavelength dependent as well. And it depends upon the power of the incident light. The brighter the source, the more fluorescence I'd see. But I can also vary other factors to make sure that my fluorimeter is within the linear range of the instrument. I can vary the sensitivity of the detectors, either the CCD or the PMT. This is a potential that we apply. The higher the potential, the larger the signal I'll see, but also the larger the noise I see. Lowering the potential means I see less noise, but I will see less signal as well. I can vary the slit widths. Having broader slit widths means that fine detail is lost from my spectra. If I have fine points, having broad slit widths means they disappear and they get lost in the whole. I can also vary the integration time, basically the length of time it takes for me to measure my spectrum. Fluorescence is an interesting one in that every instrument will give me a different fluorescence intensity. I can normalise as best as I can, but ultimately always fluorescence emission is measured in arbitrary units. Detectors are very easily saturated, and it should be checked that we're within the linear range of the instrument. Now, previously you've been taught that fluorescence is always first order exponential decay. On this figure here, that's the blue line, that is marking the first order exponential decay here. The other lines, purple, green and red, are by exponential decays. They're, again, first order decays, but I'm having more than one component. So I have two equations here. The top one is just the regular first order decay equation. The one below it, I split into two parts. I have a proportion decaying by each root, and I have the lifetime of each of the two roots separately. I see this as a variation from the first order decay, and I can change the proportion and the lifetime components independently of each other. Fitting these decays can be very difficult, and ultimately I would never try and fit anything more than a 3-component exponential decay. If there are more components than that, the probability of me being able to fit this by hand or even by using mathematical models is very small. Simple steady state fluorescence is actually quite a powerful technique all on its own. An analysis called a scattchart method can be used to determine the binding parameters of a dye molecule to either a protein or to DNA or to another molecule. In this case, I've used DNA as the example. The reason I've used DNA is because some of you might have done the third year experiment where we measure the binding of a dye to DNA. Essentially, it looks at the change in emission when we bind a molecule to DNA. It's assumed that there will be an enhancement of emission upon this binding. And so we see this increase of fluorescence intensity. With a nominally easy piece of maths, it's quite easy to derive a number of properties about the system, including the binding constant and the number of binding sites per base. From this, we can start to tell things about whether a molecule is intercalated or externally bound, or even bound to the outside of the system, because intercalation, I have only a maximum of 1 binding site per 4 bases. This can be quite a useful technique. It certainly is a simple experiment to do in a lab because a fluorimeter is quite a common piece of equipment. Something else I can do is measure the fluorescence anisotropy. The fluorescence anisotropy is another simple measurement that I can do on a steady state fluorimeter, but I can also build it up into a time resolved technique. I put 2 different polarizations of light into my sample, parallel and perpendicular as I'm defining it, and I look to see about the amount of light which is coming out parallel and perpendicular after it's passed through the monochromator. This can tell me all sorts of information about either the change in the emission transition dipole moment, or it can be used to look at the tumbling of molecules, the rotation of molecules. Small molecules such as dye rotate too quickly, and so their anisotropy is lost very rapidly. However, large molecules rotate much more slowly, and we can study this tumbling of them in solution. This is particularly used with proteins, but again has been used with DNA. The final variation on fluorescence spectroscopy I spoke about was excitation spectroscopy. This is where we fix the wavelength of the emission and we scan the excitation. We look how that emission varies with excitation. Now we would expect to see changes in the intensity of the emission due to changes in the amount of absorption of the molecule because of changes of extinction coefficient at different wavelengths. Here I have a FRET system. I have a donor and an acceptor. According to FRET, I can see this energy transfer depending upon the overlap of the emission of the donor and the absorption of the acceptor. If I look at the wavelength I've marked here, it's outside of the region where I expect any emission from the donor. I'm just looking at the emission of the acceptor. And if I scan the excitation spectra, I would expect to only see the signal from the molecule that I'm looking at the fluorescence of, the purple molecule in this case. However, if there's energy transfer in this system, I can see a component where I see absorption in my donor molecule, ultimately leading to energy transfer, the acceptor molecule ending up in an excited state, which can then emit a photon. This is a really easy and quick way to work out whether we have energy transfer within a system, and can be very useful, particularly when looking at systems involving DNA, where looking at energy transfer and reactions within the DNA could potentially be very interesting. My research focused on looking at DNA damage, and I was very interested in looking at how energy transfer occurred within the DNA. Hope this has been helpful. I'll be back with more if you want it.